Okay, this video is to give you a bit of introduction and information about the influenza antibody paper that we are going to read in this class. So, there is a wiki on influenza that you can look at for more details about the virus. I'm just going to go over kind of what is essential for understanding this paper. So, first of all, you're going to see lots of H1N something names. And so this is how influenza is named, and it tells you which hemagglutinin and which neuraminidase gene, basically, the virus has. So hemagglutinin and neuraminidase, as you can see, are the spikes on the outside of this enveloped virus. There are 18 different hemagglutinins and 11 different neuraminidases. And the 18 and the 11 mean that our immune system responds to each one of the 18 or each one of the 11 differently. These are the main parts, since they're on that outside surface of the virus, that is going to trigger antibody response or your immune response. So these are the most antigenic parts. And so our body is going to make antibodies to these parts of the virus as it makes a protective immune response. Influenza is an RNA virus, and like most RNA viruses, it can mutate. And we call this antigenic drift or antigen drift. So you get mutations, you get slight changes, but it's not enough to make an immune response not work against it, I guess. So it doesn't change the antigenicity necessarily of the virus, and so it still looks pretty much the same on the outside as what it started with. And so if you already have a protective immune response to this original virus, you're also going to have a protective immune response to one that has just had the drift. The bigger issue is antigen shift, okay? And so here we have the two input viruses, H1N2, H3N1, and we get new H and N recombinations. Or I should say new combinations that are due to what we call recombination. So if you look up here, oh, this doesn't, 4, 5, 6. Yeah. Influenza has, 8 different, hold on, does that, yeah, 8 different RNA segments. Okay, so they encode different genes. Like I said, this is on the wiki. And so if two different viruses infect the same cell, you can get new combinations of the RNA genomes, which can give you new strains of the virus. So now we have H3N2 and H1N1, as well as our original H1N2 and H3N1. Okay, so these guys are going to look very different to our immune system than the original viruses. And this happens primarily in pigs. Okay. So influenza, the reservoir for this virus is aquatic birds. Okay. And every once in a while we get a zoonotic effect where a bird influenza strain is able to jump to humans. Okay. Normally, without undergoing, like it says, change, Bird influenza does not infect people. Okay. Pigs are a host for both human strains and bird strains. And recombination can happen in pigs so that we get a totally different, here it is. Oh, look, that's so nice, I draw all over it. So we get a totally different combination of the neuraminidase and the hemagglutinin antigens, and this can produce a virus that can cause a pandemic. So a pandemic is infection that is spreading over continents, right? So these new combinations can look very different to what our bodies have seen before, and that's when we have worrisome, like the H1N1 flu that was supposed to come around and pretty nasty a few years ago. So antigenic drift is an issue for seasonal flu. Antigenic shift is a bigger issue. Antigenic drift is happening all the time with mutations. Antigenic shift is usually in the little pig. And so flu vaccines, you probably know that you're supposed to get a new one every year because the virus changes enough that the vaccine from the year before may not work against the new strains coming around. That's even true for the antigenic drift. Again, the bigger issue is antigenic shift. So a huge goal in influenza research is, can we design a vaccine that is going to work, that's going to be longer lasting, meaning it's going to work through multiple seasons. So it's going to work against and protect against all these changes with drift and shift. So that's kind of a big goal. There's lots of researchers working on this. A little bit more background. So there are 3 types of influenza. A is the one we've been talking about. Okay, so animal host, human, bird, pig, horse, and dog. We don't really worry about those too much. These can cause epidemics and pandemics. And yes, they are part of the flu vaccine. Type B is human only. And you're going to notice that we don't talk about type B with the H1N1. It doesn't mutate as quickly and usually doesn't cause pandemics because it's just antigenic drift. Type C we don't even worry about, so we don't do worry about vaccines for influenza C. Here are just a couple sites, quick reads about naming influenza and about the reason we need vaccines, different vaccines each year. Okay, so your paper is going to talk a lot about different influenza A subtypes. So here are the hemagglutinin subtypes, and you can see they're separated phylogenetically into group 1 and group 2. And I want you to see that influenza type B is way different than A group 1 or A group 2. So you might keep a little chart somewhere because as you can see, these are not in any logical order, but the paper is going to talk a lot about, is it protecting against influenza A group type 1, type 2, influenza B. So we want a universal vaccine that works against all of these different types of influenza to provide protection. And this is part of figure 1. So here, wherever you see H1 and N1, these are all type A. It talks about where it was first isolated, what year it was isolated in. I haven't been able to figure out all of these different numbers, but they're specific to the strain, so the genetics of the virus. So you're going to, again, see that they're looking at antibody protection against both A group 1 A group 2, and B. And we're focusing on the hemagglutinin because hemagglutinin is what interacts with the cellular receptors. And the head up here is much more variable. That means there are more mutations that we find in the head region than in this stem region. Okay. So the head is what our body will normally make antibodies to because it's the most accessible. The stalk or stem, so sometimes in papers it's referred to the stem or the stalk of HA. Because it's more conserved, the idea is if you can make antibodies to that stem region, you might be protected against different types of influenza because the heads are the things that are changing and the stalk is staying, isn't changing. Sorry, it's staying the same genetically, so it's expressing the same amino acids. So the big goal here is, can we have antibodies that have cross-reactivity with different strains of influenza? And this is just showing you influenza. HA binds the receptor. The receptor is a sialic acid. Again, excuse me. Check out the wiki for more information. By HA binding to the receptor, it initiates infection. So if we can get antibodies to target the hemagglutinin, we might be able to prevent infection. And so one of the goals is to find what we call neutralizing antibodies. And just so you know, AB is how I abbreviate antibody, or sometimes I just draw the structure, which you'll see in a minute. This is a pretty cool, about a 3-minute YouTube video about influenza infections. Let's see, what else did I want to say up here? Okay, so neutralizing antibodies. The goal is to neutralize the virus so that you prevent a productive infection. Human antibodies have this structure, so this is why I draw it like this. They have light chains and heavy chains. These are encoded in your genome. This top region is called the FAB region for fragment antigen binding. So you can see there's also these variable regions, and so these are what specifically binds the different antigens for a foreign substance. So these parts are what's going to bind to, like, the hemagglutinin, say, of influenza. The FC region is for fragment crystalline or crystallization. What's important about the FC is the FC stimulates the cellular component of the immune system. So it can interact and stimulate things such as T cells. So these regions are going to be talked about in the paper. Excuse me. Oops, sorry. I'm sticky. All right. And just so you know, we produce different types of antibodies. Antibodies are made by your B cells in your immune system. And the paper is going to talk about IgG. This is the main antibody in in the blood and makes up about 80% of all antibodies. So you don't have to worry about the other types. I just want you to be aware that they're going to talk about the Fc region of the IgG antibody. Something probably very new to you is the paper is actually going to use antibodies from llamas. Llamas make what we call single domain antibodies. And these are really small. So this is about 15 kilodaltons protein size, whereas our conventional mouse-human antibodies about 10 times larger. These are very stable. They are small enough genetically to be expressed in bacteria, so we can grow large amounts of these antibodies. They only are producing the heavy chain. And because they're so small, they might be able to target otherwise inaccessible epitopes. So let's go back and look at our hemagglutinin, right? So it's on the outside of the virus here. So where are these antibodies going to go? They're going to go right here. Oops, I'm having a hard time drawing. To the outside. If we want to target this stem or stalk, we might need something slightly different, because you can see there's physical steric inhibition to targeting that stalk domain. So the idea is, can we make these smaller antibodies that target something that is conserved in influenza? They then take these single domain antibodies and they genetically combine them. Okay, so they're doing molecular cloning to make multi-domain antibodies. And then they take some of these multi-domain antibodies and they put on the Fc region of IgG so that it will look more like a human antibody and trigger the cellular response. This guy, CR9114, is a human antibody that was isolated from vaccine work. Not from this paper, but it's a standard or control. And it's kind of the gold standard for an influenza neutralizing antibody. So they're going to compare their newly generated antibodies against this control. All right, so figure one, like I said, here's all these different flu strains, and they are calculating an IC50. IC50 means inhibitory concentration. So it's telling you how effective the antibody is at blocking virus infection. Okay, so you're going to see here they're using some of their single domain and some combinations of single domain antibodies. They're calculating IC50, so the inhibitory concentration of the antibody. The lower the concentration, the more effective that antibody is. So it means it takes less antibody to block the virus from infecting. Up here, when it's greater than 1,000, those antibodies aren't working against those particular strains at all. Okay. Figure 3 is also a neutralization test to see how good the antibody might be for protecting infection. So let's talk about neutralizing antibodies. Again, the goal is to block the infection. So, this can happen at different parts. So one of the reasons I had you learn about a virus replication cycle is neutralizing antibodies can block receptor binding at various points of the replication cycle. So it can block receptor binding, or maybe the receptor and the virus still interact. It might block entry. It might block uncoating. The antibody might interfere with some part of biosynthesis. Usually not. This is usually for drugs. But it also could affect the release and maturation or maturation of the virus. Okay, so again, take a look at the wiki about influenza. We don't really care where the neutralizing antibody works as long as it's blocking virus production or new infection. So there's a neutralization assay. So how do we tell if these antibodies are working or not? So the way this works is you mix your influenza virus and your antibody you're trying out. Okay. And you let them sit together so that, as shown here, antibodies can bind to the virus. And then you take this and you put it on cells. Okay. So you infect cells. Okay. And all these little circles are representing different wells. So you're doing a series of dilutions. Okay. You wait some time, you let infection happen, and you harvest to see how much virus has been produced. Okay. And you're going to do a hemagglutinin assay, which is on the next slide, to see how much virus was produced. So a totally neutralizing antibody, you would get no virus coming back, and so your hemagglutinin assay would be negative. So the hemagglutinin assay works this way. Influenza can bind to red blood cells. So when influenza binds to red blood cells, the red blood cells all clump up, and in that little well, in that little dish you're looking at, you see this red color because it's just a whole bunch of red blood cells clumped up together. So they're staying kind of taking over that solution there. Okay. Red blood cells alone will just basically pool at the bottom. So that's what you're seeing. So this is a top view. Okay. If they're clumping, they're floating all over the place, so that's why it looks red. And if you have, okay, so let me back up. So what we're doing here is we're seeing how much virus was produced. How much clumping do we get? And we figure that out through dilutions. Sometimes they do a hemagglutinin inhibition assay, mixing the virus and the antibody like we did in the previous slide, and seeing if you get clumping or not. That's not exactly what they're doing here, but it's another way to assay neutralization. It's kind of a quick and dirty way, but by actually infecting cells, you're getting a more accurate, information about the antibody. Okay, and remember from this series of dilutions, they're getting an inhibitory concentration, and the lower the IC50 value, the greater the inhibitory activity of the antibody. And 50 stands for 50%. So what is the concentration that blocks 50% of the virus from being infectious? Okay, Figure 2, don't get stuck in the crystallography methods. Don't get bogged down in how they determined which SD antibodies they wanted to use. They talk about phage display and all these series of screenings, and I've read this paper numerous times and discussed it with in other classes, and we haven't ever been able to really figure it out. That's not really the most important part of the paper. What I want you to focus on is where these different antibodies, single domain antibodies, actually bind to the hemagglutinin. That's kind of important as we think about how effective these multi-domain antibodies are. So the paper does a lot of in vitro assays, and then they do some in vivo. And one thing I want to remind you, when you're writing the words in vitro and in vivo, they need to be in italics. Okay, so they're going to use mice, and they're going to inject the antibodies that they've made and see if the antibodies protect the mice from a challenge with the virus. So a challenge means we take these mice, and the reason he's upside down with this little pipette is they do an intranasal. So they're going to squirt some virus in his nose and see if the antibodies are protecting the mouse. In this paper, they talk about humanized antibodies. Okay, humanized antibodies means that these are antibodies from a non-human species, say mouse or rabbit antibodies or llama antibodies, that have been modified to look more like a human antibody. Okay, so that's all it means. We're taking antibodies from one source and we're changing the genetic sequence a little bit so that they look like they should work in humans. So as you look at figure 4, it's doing this first assay to look for in vivo protection. I have posted the supplemental material. That's what all the S's stand for. I would take a look at these figures. They're monitoring the health of the mice. They're looking at viral load versus the control antibodies. So this paper is not super long itself, but then there's 25 pages of supplementary material. So I'm highlighting some of the figures you'd probably want to look at. And to do this, they're looking at percent survival. So they're giving the animals a lethal dose or very high dose of influenza to see how well the antibody protection works. So I just want to point out here these survival curves, even though they don't look like curves. You all start out with 100%. Here's all your animals. And every time an animal dies or has to be euthanized, your line is going to drop. Okay. So pay attention to the different concentrations of, in this case, antibodies, of this case, virus we'll talk about in a minute, AAV. Vehicle is another control. So if you do something to your sample animals, you have to do the same thing to your control. So instead of injecting intranasally giving them a squirt of virus, they're just going to give them a squirt of saline solution. So that's the vehicle. Whatever they've resuspended the virus in, they're going to give that same liquid without the virus to their controls. This part of the graph is focusing on a different delivery method using a second virus called adeno-associated virus. And again, this is the second half of figure 4. Here's some supplemental. And what they're doing here is instead of injecting the mice with antibodies, they are giving the mice this adeno-associated vector that contains DNA that codes for the multi-domain antibody. Okay, so the multi-domain antibody is going to actually be produced in the mouse. Then they do that same kind of challenge and look at survival curves. So AAV is a very common virus used for, it was looked at a lot for gene therapy. It's a nice little small virus. It's non-pathogenic. It comes into our cells and does a good job at expressing genetic information. You don't have to worry about all of this information. The nice thing is there are multiple subtypes, so multiple strains of AAV that look slightly different. So the one they talk about in this paper is AAV9. The reason that can be important is because, just like to any virus, you can produce antibodies. And so you don't want to have antibodies that's neutralizing your gene delivery method. And so they mention that in this paper, even though mice, some have AAV antibodies, that the delivery system was still pretty effective. So I think that should get you going on the paper, and then in small groups you will discuss the data and the conclusions and the implications from this paper. All right, thank you.